Ki te tangi e te manu e karanga nei tui, 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 tui. Tui e te runga, tui e te raro, tui e te roto, tui e te waho, tui e te hera tangata. Ti he Māori ora. Ena mana, ena reo, el ra rakatira mā tēnā koutou, a tēnā koutou katoa. A ko Waiao, ko Richard Blakey, taku ingoa, no Otapoti hao, ko Katarani te iwi hei kamaihia ha te whare wānanga o tākou, tēnā koutou katoa. Kia ora everyone, and welcome to this wonderful celebration tonight for the presentation of the Carl Smith Medal to Associate Professor Peter Mace from the Department of Biochemistry. My name is Richard Blakey. I'm the Deputy Vice-Chancellor for Research and Enterprise, and I am here with the academic party to preside, and so we have with us Professor Sally McCormick, the Head of Department, tēnā koe. We have apologies from Professor Helen Nicholson, the DVC academic, who is unwell this evening, but we are very privileged to have our Vice-Chancellor, Professor David Murdoch. Tēnā koe o te tumuaki e te wharawānanga o Otago. It's our privilege to have you here, David, who will be presenting the Carl Smith Medal to Peter after his lecture. So it's a wonderful celebration. It's my great pleasure to share this celebration of outstanding research achievement with you all, friends and whānau of our medal recipient, members of our university community, citizens of Dunedin in the region, and people zooming into the live stream from around the world. So hopefully all of the AV is working well for this. So no mai, haramai, welcome. A particularly warm welcome tonight goes to Peter's supporters and family. Special greetings to your wife Gabriel and daughters Rita and Nell, who are here tonight. Welcome. I hope this is a fun night for you all. And to your other whānau who are online. Hello to you all. Also greetings, this is an international event, to your collaborator James Murphy, who is visiting from Australia, and other departmental colleagues. And I understand that there's going to be a nice celebration with some of your family and colleagues after the event. Enjoyable duties that I have the privilege to perform in my role each year. And I both oversee the selection panel, but this is the ceremonial part that's much more fun, because the selection process for these awards is quite grueling given the level of competition. Now, the order of ceremonies is that I will say some words, and then I'll invite Professor McCormick to introduce the lecture. Following this, I will summarize the significance, give a vote of thanks, and then let's say, provided the lecture is suitably impressive, so this is a test, Peter, I will then invite the Vice-Chancellor to present you with the award and the medal. That will end the formal proceedings, but we'll also ensure that your achievements are recognised when you are presented again with a certificate at the University Staff Awards function on Wednesday. Before I give you some background, I just wanted to highlight as well that this is coming at a very timely process, because we have just launched our annual research highlights publication for the university, Hei Ki Tinga. And in fact, this year's edition is entitled Talented Futures. There are a few editions here, perhaps for your family to take if they wish to first. And Peter's award is recognised in this official document of the university. It's particularly nice because we have focused on early and mid-career researchers in this edition, and this Carl Smith Medal is specifically awarded for people in the earlier stages of their career. For those of you who cannot get a copy, or for those of you viewing online, it is actually available online also if you search Hei Ki Tinga. Before I ask Sally to introduce Peter, a little about the award and the medal he will receive and the names. So why a Roheath Trust Award and a Carl Smith Medal? Well, one is for the money and one is for the glory. The Roheath Trust was established in 1964 to support the work of the university, and it was established by Carl Smith. So Carl Smith and Roheath Trust are intimately linked. Carl Smith was a prominent Dunedin businessman and a member of the University Council. And he endowed, through the Roheath Trust, a $5,000 research support award that bears the Trust's name. At the same time, the university commissioned the Carl Smith Medal in his honour. And in keeping the Carl Smith name, it gives the university a good opportunity to recognise the contribution of Sir Carl and his wife, Lady Catherine, that they made over many years after coming to New Zealand from the family estate of Roe Heath in Birmingham. And the connection of Birmingham is to the Cadbury empire. So that family connection to the university and the ward continues. And until very recently, Sir Carl's daughter Catherine, who grew up in Dunedin and spent her golden years in the Otago region, was a regular attendee at these awards. Now, Sir Carl was also a well-known author in addition to his business and community contributions. He was known not so much for his book Sweet Success, the history of the chocolate maker Cadbury Fry Hudson, published in 1968, but also for a quirky and humorous tome called New Zealand from N to Z that has become somewhat of a collector's piece for its interesting insights into mid-20th century New Zealand culture and society. Now, the book is long out of print, but one of the things that we will present to you, Peter, tonight is a rare and hard to come by edition of the book. It will be unique because there are ongoing and active searches on Trade Me and other sources, and the Regent Book Fair gets scoured every year to try and find some additional copies. I think we're down to 2 or 3 left, but you will receive one of these rare editions as well as your medal, as well as your prize. So look, this is not just a double award, it's a triple award. So anyway, that's the order of proceedings. We'll get Sally up, then we'll have your lecture, and then we'll present your award. So without further ado, please accept our congratulations on behalf of the university. And I'd now like to invite Sally, as Peter's head of the department, to introduce his talk to you. Nō reira, tēnā koutou, tēnā koutou, tēnā koutou katoa. Thank you.
Tēnā koutou katoa. He pai kite kia koutou. No mai, haere mai. Ko Sally McCormick tōku ingoa. Welcome everyone. It's really good to see you here tonight. It's a bit of a crowd here tonight. Warm welcome to Peter's family, his colleagues and his friends who are here to support him in delivering his Carl Smith Medal tonight. I'm Sally McCormick, Head of Biochemistry, and I have the great pleasure of being able to introduce Peter tonight. So just a few things about Peter's background before I tell you a little bit about the lecture. So Peter is an Otago graduate. He's remained very loyal to Otago. He completed his BSc Honours in Biochemistry in 2002, then stayed on to do a PhD in the department with John Cupfield, and then further stayed on to do 3 years worth of postdoctoral studies in the lab of Catherine Day before moving to the US to the Sanford Burnham Medical Research Institute, where he took up a postdoctoral fellowship there in 2010. He returned home in 2013 on a prestigious Rutherford Discovery Fellowship and subsequently went about building his own laboratory. He actually won the Early Research Career Award for Distinction in Research at the university here in 2016. And in that same year, was promoted to a senior research fellow. And the following year, he was appointed to a senior lecturership position in the department. He was promoted to associate professor in 2020. And this year, he was appointed as our associate dean of research for the School of Biomedical Sciences. A long list of achievements in a very short space of time. Over the course of his career, he's established himself an international reputation as a leading structural biologist. This means that he solves structures, in his case, 3-dimensional structures of proteins. This is important because proteins are the workhorses inside our cells. They tell our cells how to respond to our environment. They tell our cells whether to die, live, or multiply. And by understanding the structure of proteins, we can learn a lot about proteins. The active parts, their partners that they interact with, and what switches them on and what switches them off. And also the effect of mutations that there might be in these proteins. Peter has solved, I think, I've lost count, but it must be something over 20 structures, I believe. And in doing so, has taught us how proteins talk to each other to facilitate cell signaling, But his work actually has a lot of implications around drug design, because proteins are increasingly becoming targets for drugs. And so if you can understand how proteins interact with these drugs, you can make for a more effective design. He's been very prolific in his publishing, over 50 papers, invariably in very high quality journals, such as a number of papers in the Nature suite of journals, which are among the highest quality journals in science. And this is no significant feat in a field that requires a lot of experimental work to actually get published. He's highly cited. Several of his articles have been chosen as cover articles, which means the editors think they are of really broad interest to people. He's highly sought after as an international speaker, and has just been elected president of the New Zealand Society for Biochemistry and Molecular Biology. Probably means more conference organisation, I suspect. I just wanted to finish up by just making a comment about Peter as a person. I recently saw a Maori proverb, or whakatoki, which I think plays to a couple of key properties that Peter has, which makes him very special. And the whakatoki goes, kaore te kumara e korero mō tōna aki rika. And that translates to, the kumara doesn't speak of its own sweetness. And what it's really saying is it's about being humble, and it's about ensuring the success of others. And I think Peter has done a great job of that. Richard already sort of alluded to it in his words. Peter has made, and there's a couple of examples, Peter has made sure that his collaborators and his colleagues get access to all of the latest technologies in structural biology. This is seen in his huge efforts to access both the Australian Synchrotron and the cryo-EM facility in Australia, organising visits there for his collaborators, and also through the way he trains his students. He's a very careful and considerate trainer of students, ensuring their own success as well as his own. So we are very lucky to have someone so collegial in the department and so successful in the department, and we're just happy that Peter calls biochemistry his home. So with no further ado, I'm going to now hand over to Peter to talk to us about his structures, his switches, and cancer signalling.
That's going. Loud enough. Kia ora koutou katoa, no uropi o kutipuna, no tokoroa ahau, e mihi ana ki ngā tohu o nehe o Otipoti e noho nei au. Ko Peter Mace toko ingoa, no reira tēnā koutou katoa. So thank you very much for the invitation, this opportunity to speak to you today. It's great to see so many familiar faces, family who've helped all the way along, colleagues who I've worked with directly, colleagues who have helped just by being there, and family online who are watching. So it's really exciting to be able to talk to you about some of our work today. So my talk today is entitled Structures and Switches, Cancer Signalling at the Molecular Level, which will hopefully become clearer as we go along. But what we're really interested in my lab is thinking about how a cell communicates within itself, and so how it makes those decisions that Sally's been talking about, how it decides whether it should be growing, whether it should stop growing, whether it should change into another cell type, because when those processes go awry, that's when we get diseases such as cancer. And so where we're going to start is this picture of a couple of cancer cells. And so these are HeLa cancer cells. So this picture is taken with an electron microscope, looking at the surface of these cells. So HeLa cells were derived from a cancer patient called Henrietta Lacks, and they actually have underpinned pretty much all of our modern human cell biology, because these cells were the first cells that we learnt to be able to grow in the lab. And so the reason we were able to do this is that these cells were basically immortalized. So they will just keep proliferating even when the conditions aren't quite right for them, they'll keep proliferating. That enabled the early researchers 50, 60 years ago to work out techniques to grow cells in the lab, and that's underwritten basically all of the work that we do in cell biology now. But underwriting that is the fact that they are immortal. They've lost that communication inside that tells them we should stop proliferating, and we need to stop growing at that stage. And so that's really what my lab is interested in, what's happening inside the cell that is allowing it to communicate and realize this is what we should be doing, and when that process goes wrong, why we get diseases such as cancer. So this is looking at a signaling pathway in its probably simplest form that we can imagine. The cell might receive some sort of signal. It might touch another cell, or there might be a growth factor that comes along in the blood, or it might be something that happens within the cell. This turns one protein from its off form to its on form. So we're going red to green like a traffic light here. What that protein can then go and do is go and turn on another protein. And so this is a very simple signaling pathway. There's just two components, but in cells, this can have multiple different steps and multiple different kind of crosstalks, which we'll talk about. Once that protein is on, it might go off and do any number of different things. So it might go into the nucleus of the cell where it can control what genes are being expressed, turn things on or off. It might tell the cell that it should move away and become motile and move to a different part of the body, or it might go off and turn other proteins on and off on the cell. So it might tell the signaling pathway, hey, we've got enough signal here, let's turn it off. And so understanding these signaling pathways obviously gets more and more complex when we think about all of the things that's going on in the cellular environment and the things that cells are experiencing. So they have to integrate a huge number of different signals that are telling cells whether they should move, telling cells whether they should change into a different cell type or proliferate. And so even in this relatively simplified form, things start to look relatively complicated. When we've got a nice clean picture like this, though, we could think of this signaling within a cell as something like a wiring diagram for the physicists amongst in the audience. Unfortunately, whereas a wiring diagram is relatively clean, what we're dealing with in cellular signaling is much more something like this. So it's a system where it's probably functional in some sense, but there's a lot of things going on there that we don't really understand yet. We don't quite understand what's connected to what, and within that whole kind of mess, there's probably some things that are broken or may become broken, and so diagnosing how that's going wrong it gets really quite complicated. And so what we're trying to understand is when these switches go wrong, how does it change the communication within these signaling pathways? So one of the ways that proteins communicate and get switched on and off is this process called post-translational modification. So the post-translational, that you don't need to worry about that too much. That's just talking about after the protein is made. So the protein's sitting around inside the cell. It can be turned on by lots of different things, but two of the main ways that proteins are turned on or off are these processes called phosphorylation. So a phosphate atom gets put onto a residue within the protein, or you can have another protein get put onto the protein called a ubiquitin molecule. And so both of these things, the important thing about a signal like this is you have ways that it can go onto proteins and ways that it can come off, because if you have a signal stuck on, that's not necessarily going to be a very good way of controlling a cell. You want reversibility. And so there's proteins in the cell that write those modifications onto other proteins, and there's proteins in the cell that can erase those modifications. And so in this kind of simplified drawing, I've shown you that they turn on or off proteins, but obviously proteins are more kind of complicated than these jelly beans that I'm showing you here. And so what we really need to do to understand how those phosphorylations and ubiquitinations can turn on and off proteins is understand them at a bit more detail. And so, as Sally said, what we spend a lot of our time doing is trying to understand what the three-dimensional structures of proteins are. And so this is kind of on the left-hand side is the jelly bean that I've already shown you. On the right-hand side is a closer representation of what a protein structure really looks like. And this is a relatively well understood, relatively simple protein, but it looks pretty complex because it's a kind of a compilation of hundreds or thousands of atoms, all sitting in relation to each other. So understanding exactly their 3D positions can be really useful, which I'll show in a second. Even people who spend all day looking at protein structures, that's not a very decipherable picture. So often what we do is look at protein structures in this kind of cartoon form, which looks a bit more attractive in the middle. And so that's what I'm going to be showing you. Most of the protein structures are going to be in that cartoon form that we look at today. So how do we actually solve that structure which you're seeing on the right-hand side there? So we use this process called X-ray crystallography as one of the ways we solve protein structures, and up till recently has really been the predominant way that we solve these three-dimensional structures. And so X-ray crystallography, as the name suggests, at the start of it, we need a crystal to start off this process. And what is shown on the left-hand side here is a crystal of a protein. So this is one of those things that has hundreds or thousands of atoms. If we incubate it in the right chemical conditions and just happen to have the right concentration of protein and all sorts of different chemicals, and you're standing on your left leg and you scratched your right ear at the right time, you might get a crystal like this grow. And so this can be quite a trial and error process. But if this happens, what you then have to do is get something that's about the size of the eye of a needle, or probably smaller than that, fish this crystal out, and after probably smashing a few of them, you can freeze one of these crystals and then shoot it with a really intense X-ray beam. And so if all these stars align, what you might get is a diffraction pattern, which is those spots in the middle of the slide there. And through a bunch of maths, what you can then do is calculate a three-dimensional map of where all the atoms in that protein actually sit within that crystal. And so then you can then build a model of where all the atoms within that map lie. And this is pretty much like fitting a puzzle. When Nell was younger, she used to tell me about talk about how I was doing my puzzles, Dad, which is kind of pretty much what it is. It's pretty fun if you have an electron density map like this, fitting a model in there. And so this process can be quite involved, and it really involves quite a lot of technology as well. As I alluded to, we have to set up, this is quite a trial and error process, growing protein crystals. So we use this robotics to set up tiny, tiny trials. We also use this imaging system, which is our rock imager, to take pictures of these crystals as they go along. So we've had really good support from the Large Equipment Fund at the university in helping us purchase this imaging system, and it's been really a boon for the area of protein crystallography at Otago, and also support from Lottery's Health for both the robot and the imaging system has helped us along the way. And then to shoot these these crystals, what we have to do is take them to the synchrotron in Melbourne. And so this is a source of really powerful X-rays, and it basically allows us to solve crystal structures that we wouldn't be able to solve using any X-ray source in New Zealand. So we get good support from the New Zealand Synchrotron Group, which Otago is a member of, to be able to go and both visit on site and train students at the synchrotron, or just send our crystals there and shoot them. So before we even get to that stage though, there's just a huge, it's hard to kind of undersell how much work goes into making the protein to be able to grow those crystals. So there's just huge amounts of work that go on from staff and students in the lab. Not all of it involves probably quite unsafely crawling underneath our FPLC machines there. But basically I'm going to show you a few structures later on in the lecture, but really there's just huge amounts of work that go into even being able to make the protein in the first place, let alone convince it to form crystals. And so it can be a huge amount of work to solve a crystal structure. So why do we go to all this trouble? And so as some of you will know, one of the most obvious ways that we can use these three-dimensional structures is to be able to design better drugs. And so just to illustrate this concept, quite often what a cancer cell might do is it might pick up a mutation in a gene that encodes a kinase. And so a kinase is one of those proteins that puts it's one of those pencils from the slide earlier. It writes a phosphate modification onto other proteins. And so what this can do is, if this mutation basically turns on that kinase and makes it go and phosphorylate things kind of out of control, then you can have a situation such as you get uncontrolled cell growth, the accelerator's stuck on and it'll just keep proliferating out of control. But if we can solve a three-dimensional structure of that kinase protein, what we can do, this is then a template, it's kind of knowing the structure of the lock to be able to fit a key in to design a drug to sit in the active site of that kinase protein. It stops it from being able to bind the molecules that it needs to bind to phosphorylate things, and so that's an effective way to improve drugs by knowing exactly how they fit in 3 dimensions inside the kinase protein. But that's not, we do have projects in the lab that are doing this sort of thing. That's not really what I want to talk to you about today. What I want to talk to you about is more how we can, a bit more holistically, use solving protein structures to understand more about how cells work, because that's going to kind of bring the camera back a little bit and think about what areas we might be able to develop drugs in the future, understand where things are going wrong in cancers that we already have, and understanding whether we can target particular protein switches in a better manner. So these are the 3 examples I'm just going to give you some brief details about for the rest of the lecture. And so the first one starts off thinking about taking a drug that we already know is really effective. It's been used for decades, but it was not really understood what was happening downstream to connect to signaling pathways that made it effective. And so this story starts, like all good stories, in Madagascar. So on the left-hand side there is a Madagascan periwinkle. And if you extract compounds called vinca alkaloids out of the leaves of these, the Madagascan periwinkle, you have drugs which are really have been used for decades and decades, called vinblastine and vincristine. And so these are really effective chemotherapy agents. They are on the WHO list of essential medicines. And the way that these work is they work by disrupting the cellular cytoskeleton, and in particular these things called microtubules inside cells. And so they're stained green in the right-hand side there. So what microtubules do is all sorts of kind of mechanical work around the cell. So they might move proteins and vesicles around. They might do things like draw chromosomes apart when a cell is dividing. And so they're really integral components to how a cell can function. But when you treat cells with things like vinblastine and vincristine, what happens to microtubules is you get a proliferation of these kind of curly ends that you might be able to see. We've got a bigger picture in a second. But basically, most of the microtubule is straight, but at the ends it has curls. And if you treat with drugs, these particular drugs, you basically get these kind of curly things over proliferating, and you've disrupted your microtubules from being able to form in a nice way. One of the proteins that it was known to be important for the efficacy of, how well these vinca alkaloid drugs work, was this kinase protein called MEKKI1. And so this is a kinase. As I said, kinases put phosphate onto other proteins. And what MEKKI1 does is triggers this kinase signaling cascade. So one kinase activates another kinase, which activates another kinase. And the downstream effect of this is cell death, which is what you want in effect and when you're dealing with a chemotherapy. But it wasn't really, there was no logic behind why MEKKI1 was an essential kinase for the efficacy of these vinca alkaloid drugs. So MEKKI1 had a kinase domain, which is shown in the middle there. It's actually quite a large protein though, so it had these extra domains which we didn't really know what they did. And so our approach was to look at these other domains and try and understand how they might be connected to this microtubule disruption that vinca alkaloids carry out. And so a PhD student in the lab, Pavel Filipchik, came along, and one of the first things he did in his PhD was to solve the structure of one of these additional domains of MEKKI1. And so that's what's shown on the left here. So this is a domain that was of previously unknown function. No one really even proposed that there was a domain there. He solved the structure, which looks like it does there. It looks kind of like a bunch of squiggles lined up next to, spaghetti lined up next to each other. So that didn't tell us its function first up. But one of the cool things when you solve a protein structure is you deposit it to this place called the Protein Data Bank. And so there's, I think, almost 200,000 protein structures that are deposited in the Protein Data Bank. And so what Pavel could do is search his structure against all of the other protein structures that people had solved. And what he found is it's actually a really close match for this protein that's shown in pink here. And so that protein in pink happened to be a protein that people had studied really in quite some depth, and it's actually a protein that binds to microtubules. So it's a tubulin binding protein. So tubulin is one of the building blocks that makes up microtubules. So it looks like MEKK1 might have a direct domain that can actually sense and bind to these microtubules. Little bit more background about tubulin before showing the next step is that to get the straight part of tubulin versus the curly part of tubulin, tubulin has like a straight and then a more open form, which it's just kind of opening and shutting in that little movie there. What these vincristine and vinblastine do is they actually, I've got to time it right here, they wedge in between those two domains of tubulin, and so they stick it in that kind of more open form. And so that's why you end up getting these kind of curly forms, because those vinca alkaloids are getting themselves stuck there, and it builds up these curls. So it becomes a pertinent question: does this domain of MEKK1 bind to the straight part of the microtubule or the curved part of the microtubule? And it turns out that it actually binds specifically to the bent part of the microtubule, which is induced by these vinca alkaloid drugs. And so we now know that MEKK1, which there was previously no reason for it to, well, no logic to why it was activated by microtubule disruption, actually has a domain which gets it to these curly tips of microtubules. What we could then do is actually look in some tumour sequencing databases. So patient tumours had been sequenced, and find that there's actually a couple of hotspots of mutations in MEKK1 which sit at that tubulin binding interface. And so previously, before we had that structure, there's no reason we would have understood why those mutations keep on occurring in patients. But it turns out that these mutations, so Pavel tested some of these, actually disrupt its ability to bind to tubulin. And so that was the end of Pavel's PhD, but it's not necessarily the end of the project. So we have Alex Bowles in the lab, who's a PhD student, really trying to work out what happens after MEKK1 gets recruited to these curly ends of the microtubules, trying to understand this, like how the switching actually happens. At the moment, we've got the connection, but we don't necessarily have the switching. And so that's where the story is going now. So the second example I want to talk to you about is thinking about how, when we already know a gene and a protein is a hotspot for getting knocked out in cancers, understanding its structure is really important to understand like how these mutations are knocking out function. And so this, to understand this story, we need to have a little bit more background about how DNA is packaged. And so some of you will be aware, inside one cell of your body, you have about 6 feet of DNA. So it really needs to be packaged really effectively for it to be able to fit inside the cell and inside the nucleus. And one of the ways that a cell packages DNA effectively is by wrapping it around these proteins called histones. And so this is kind of analogous to wrapping cotton around a cotton reel. It keeps things tidy, but then these histones are packaged even more efficiently into these kind of superstructures called condensed chromatin. And one of the marks of condensed chromatin is a single ubiquitin molecule gets attached onto the histone proteins. And so this is kind of a put you away, will keep you away for later. When the genes need to be expressed, you'll have the DNA gets unpackaged, and this gives access to proteins that turn on gene expression. So a single ubiquitin molecule is a marker for basically DNA structure. The protein complexes that put on and off this ubiquitin mark then become really important for controlling what genes are going to be turned on and off at different times. And so the protein complex that we've been studying in the lab, which takes off this ubiquitin mark, is made of two main components. One of them is called BAP1, and so BAP1 is a protease, so it's a protein that can cleave other proteins, and what it does is it cleaves this, the C-terminus of ubiquitin, so it chops it off from histones. That's where the catalytic activity of the complex comes from, but it's activated by this protein called an ASXL protein. There's a few different ASXL proteins, but it needs this partner to activate it to be able to turn on its activity, so that's going to remove the ubiquitin marks and promote more kind of open chromatin structure where your genes can be expressed more actively. Now, BAP1 was known to be a really important cancer gene because of a syndrome that certain families had, which made them more susceptible to environmental carcinogens. And so there's germline mutations in BAP1 can predispose to certain melanocytic tumours and also to cancers such as malignant mesothelioma. And so mesothelioma is generally a cancer which occurs when you're exposed to asbestos, but only at really high levels if you've got kind of normal BAP1 or some other predisposition genes. But if you pick up a mutation in BAP1, then even kind of lower levels of asbestos become more dangerous. And so understanding how these mutations knock out function of BAP1 might be useful for being able to predict, is this person got a damaged copy of BAP1, will they be more susceptible to asbestos exposure or other carcinogens? So this structure was solved by, first she was a PhD student in the lab and became a postdoctoral fellow in the lab, Martina Fogliso, who I co-supervised with Catherine Day. And so what Martina was able to do was to solve a crystal structure which showed us how the protease component of this complex was turned on by this ASXL protein. And so the blue and yellow parts are the protease part of the complex. ASXL has this kind of bundle of helices here which wrap around the C-terminus of the BAP1 protein and kind of hold this end out of the way. The end result of this is a binding site for ubiquitin which sits here. It's basically a binding site that's made up by the yellow protease domain and this green domain from ASXL, which there was no real, that doesn't have a catalytic function of its own. Basically its function is to hold BAP1 in that conformation, and through this it can recognize ubiquitin really specifically. And so this is important because you don't want your protease just chopping other proteins non-specifically. You want it to be able to recognize ubiquitin and cleave it at the right time. What we could then do is use this structure to map where mutations that were occurring in patients were showing up. And so what we can see, so in this representation on the right hand side, when we have basically, when that worm gets a bit fatter, that means there's more mutations showing up, and these have gone red as well in this case. So mutations basically are clustered around that ubiquitin binding site. And so this suggests that tumours that have got these mutations in BAP1 that affects their binding site for ubiquitin, so this is disrupting the ubiquitin binding site, they get a selective advantage, and so this is promoting their growth, their uncontrolled growth. And so tumours are selecting for the most functionally important parts of this BAP1 molecule. And so that's how the BAP1 part of the complex can get knocked out, but there's also mutations that show up in the ASXL part of the complex. And so where this project is going now, a PhD student, Cameron Reddington, and Abigail Burgess, who's been a researcher in the lab for some time, really have been digging down into how this complex is targeted across the genome. And so we've understood how the catalytic activity, how that protease activity is knocked out, but which areas of the genome that's directed to is going to become really important, because that's going to turn on gene expression in different areas of the genome. And so one of the areas of interest here is that if you mutate the ASXL component of the complex, that drives leukemias in general, whereas if you have mutations in the BAP1 part of the complex, that drives things like mesothelioma and melanocytic tumours. And so the targeting of this complex seems to be leading to different types of cancers depending on how these mutations are arising. And so that's what Cameron and Abby have been doing, working out how the other domains of ASXL proteins direct this complex, kind of guide it around different parts of the genome. So that's really been about understanding how things get broken. The last part of the last example that I want to talk to you about is really trying to understand, can we target a particular protein? Would it be a good thing to spend all that effort into making a drug that fits a particular active site to block its function? And so this story is dealing with proteins called the Tribbles proteins. And so there's 3 Tribbles proteins in humans, but the way that Tribbles proteins were discovered were back in the early 2000s in Drosophila, so people who study fly genetics. What they did is created mutations in the Tribbles protein, and or the Tribbles gene, and what that did was drove kind of the uncontrolled proliferation of these brown cells at the bottom of a Drosophila oocyte. And so, of course, they were fly geneticists, so they're probably Star Trek fans. And if you're a Star Trek fan, which I am not, but I've learnt about through working on Tribbles proteins, Tribbles have quite a cult following in the Star Trek world. And so what they are is these kind of fluffy things that are, in this case, taking over the Starship Enterprise. They grow out of control. And so you've got to admit, they do look something like the cells that when they're stained like that. And so they named this gene Tribbles. And so it's got such a cult following. You can buy all sorts of, you can buy Tribbles slippers and Tribbles beer and all sorts of things if you dig deep enough on eBay. But that's not the reason that we got interested in Tribbles. Tribbles proteins are part of this really interesting family of proteins called pseudokinases. And so I've already talked to you about how kinases are the proteins that put phosphate onto other proteins. But what pseudokinases are, they are things that look like kinases, so they have the same shape as a kinase, but they don't transfer phosphate. So they've lost specific residues in their active sites that enables them to be able to do the actual kind of chemical reaction to transfer the phosphate. So they look like one thing, but they're not quite that thing, much like these dogs on the right-hand side. So this is a really interesting area of biology. It's also a really great community of researchers. So James is not looking quite as warm as he is in San Diego down there, but there's really a good community of researchers looking at these pseudokinases, trying to understand their functions, because they basically pervade lots of interesting areas of biology. And so what Tribbles actually do is they bind to another protein called COP1. And so what COP1 does is it transfers ubiquitin onto other proteins. And so I've said my lab is kind of interested in these post-translational modifications, which is transfer of phosphate and transfer of ubiquitin. And this is a thing that looks like something that transfers phosphate, but it actually helps transfer ubiquitin. So this is like the perfect project for us. But what Tribbles do is they bind to substrate proteins, which are shown in green here. What those substrates generally are transcription factors which control gene expression. And so if they put these chains of ubiquitin onto transcription factors, what this is a signal for in the cell is that we've completely got to degrade that protein. We've got to get rid of it completely. And so this is a really effective way of turning off gene expression. And it just so nicely happens, actually, protein degradation is one of kind of the most exciting areas of drug development at the moment, because you can think that instead of just trying to block a protein from doing its function, if we could degrade it completely, that gets rid of it out of the cell, and you've got rid of your kind of cancer-driving protein completely. So it's really kind of a hot area of biology, but it's also got really lots of kind of fundamental interest as well. Now, in terms of the genes that Tribbles actually regulate expression of, these genes are really highly involved in myeloid cell development and proper differentiation of myeloid cells. And so when you have too much of the Tribbles proteins, what this can actually do is cause too much degradation of those transcription factors, and that can drive acute myeloid leukemia. And so this is an example from our collaborator, Karen Keeshan, who's now in Glasgow. Basically, if you transplant bone marrow overexpressing TRIB2 into mice, all of them basically get acute myeloid leukemia. And so going in, the logic was there that if we could block this function, that would be a good thing, because too much Tribbles is a bad thing. But we have this thing that looks something like a kinase, but we don't know if the active site of that protein has anything to do with its function. So would making a drug against these proteins even do anything useful if it bound in that active site? So we've been working on Tribbles proteins for quite some time in the lab. It's probably one of our longest running projects, and a lot of that work has been led by Sam Jameson, who's a researcher in the lab. And I'm just going to summarize like 7 years of Sam's work in one slide here. But basically what we've solved a bunch of different structures of Tribbles proteins. One of the things we've discovered is that it has this form where it holds onto its tail. And this tail is basically the part of the protein that goes off and binds to the ubiquitin ligase called COP1. And so it's got an off form and an on form. And so what I'm going to do is morph between those two structures to show what's going on. And so we've also discovered how a substrate binds to TRIB1. And so when the substrate comes along, this actually causes allosteric changes through the pseudokinase domain. And this actually pushes the C-terminal tail away from the pseudokinase domain. So basically the substrate binding and the letting the tail go to go and find the COP1, those are coordinated. It's a coordinated event. But if we just focus our attention kind of on that middle part of the Tribbles protein there, what you might notice is when the substrate's on there, I'll pause it, there's changes going on in that active site, which if we could stabilize one of the structures or the other, this might mean that we can actually block function of substrate binding. So I'll just show that again. So you look, there's residues that are changing in there. If we could stabilize that activation loop in one conformation or the other, we should be able to block substrate from binding, so we can block Tribbles function. And so this is just shown next to the kinase I showed you earlier, where a kinase drug sits in the active site. So this is really proof of principle that if we can make drugs that sit in this active site, even though this is not an active kinase, it's a pseudokinase, we should be able to block its function because it's looking like it's working in the same way. So that comes to the end of my 3 examples. Hopefully I've illustrated why solving structures is useful on various different levels. So linking linking mechanism of known drugs, understanding how protein structure gets disrupted, and also understanding whether things might make good drug targets. Just on the final slide, what I want to illustrate is where we're going in the future. So as I've already shown you with that kind of movie, proteins, they are really, we show them often as static things, but really they're moving around, and so trapping them in particular conformations is really useful for understanding protein function. And so what we've been doing is starting to try and make these things in pink here called protein nanobodies. And so these are useful for trapping structures of proteins, both to manipulate how they behave and also potentially to solve structures. And so this is a really nice image that's just came out earlier in the month, made by Ben in our department of Tribbles. This is the TRIB2 structure trapped by a nanobody, and it's being kind of taken over by by Tribbles. My brain's been taken over by nanobodies. But the other thing that's really useful for trapping these flexible proteins is this an alternative technique for solving structures that doesn't require a crystal. And so I've said we spend a lot of time making protein and trying to grow crystals. We still need to spend a lot of time making protein, but if we have the right type of protein that's large enough, we can use this process called cryo-electron microscopy to solve structures directly of proteins. And this has really basically just exploded in the last few years in terms of the techniques and the technology that enable this have really changed the game and how we are able to solve structures. So it's really an area we've been leaning heavily on our Australian collaborators to be able to do this at the moment. It's kind of a little bit of hard work at the moment doing it from afar, but we're doing our best to kind of move with the times in there. So we're kind of moving to basically using any method that we can to solve protein structures to inform protein function. So that just brings me to thank all the people, the lots and lots of people who have contributed to this work. Firstly, they're not listed up here, but my family for all the support over the years and being very tolerant of when I'm not there, either physically or mentally. Yeah, the support's really appreciated. And then to my group, first up in my group, Sam and Abby really are the ones who keep the wheels turning in the lab. They keep everyone else in line. They drive their own research projects, but they also make sure everything runs really well, make coming to work just fun. And this is a list, I think, of most of the students. I might have missed a couple of names there, but we've had a really great crop of honours and PhD students. I've named kind of some specific ones along the way related to specific projects, but really all the students through the lab have been great. We've got great internal collaborations with Liz Ledgerwood, Catherine Day and Anita Dunbeer on various different projects. Some I've talked about today, some I've not had time to talk about. We also collaborate within New Zealand with Ren Dobson at Canterbury. We've got a great slew of international collaborators, James from the WeHi, who it's great to be able to have you here today. And we also work with David Croucher on the MEKKI work at the Garvan Institute from Sydney, and then a bunch of people from both the UK and the US who have been really pivotal in being able to get a lot of this work done. And of course, none of this work gets done without funding. We've had fantastic support from starting off with my Rutherford Discovery Fellowship, had great support from the Marsden Fund and the HRC in terms of actual projects. Lotteries Health and UORGS have been great for getting projects done and equipment purchased. And as I said, the New Zealand Synchrotron Group has been great for being able to get to Australia to use the synchrotron. And with that, I'd just like to thank you all for listening.
Kia ora, Peter, and thank you so much. That was a tour de force. And actually, I'm just identifying that you have fun in your work and your leisure time, and that picture from the escape room at the prison probably sums up the kind of attitude that you have to work in this area. That it's hard work, and escaping from coldits at times, I'm sure, would be an easier task than unlocking some of these protein structures. Look, I'm not going to try as a humble physicist to even try and walk us through that, but I will give some remarks. The first thing is, it was very clear and evident right from the start, your happy smile and your body language, how much you enjoy what you do and how much you enjoy communicating and disseminating that, how much you enjoy the challenge of structure, linking structure to function, understanding how that fits into the beautiful, complex, almost non-understandable nature of the way that biology works. And you gave us an example of the tour de force of that. I was very pleased that you introduced, for me as someone who's taught in electrical engineering, a beautiful simple circuit, the BC547, and a single transistor switching circuit. I thought I could follow, but at that point, then this idea that we can somehow think of simplicity in switching, but the complexity of real life is often beyond our comprehension unless we start to do the things that you showed that we can do so carefully, look at individual pieces and puzzles and try and build up things together. Your ability to communicate is very clear. The visualization and the cartoons that you produce help to guide us through that journey. And you very much then led us to three questions that not only identify issues that if we were a pharmaceutical company would stop at that first question, why is a switch triggered? And we would go to ongoing research to look at individual things and turn the handle on solving structures and trying to identify molecules that might go through a long and increasing list of targets that we're after. But you demonstrated that at a university we need to go beyond that and look at the bigger picture still, looking at the fundamental knowledge of why things work, how can these switches be triggered at a fundamental level. What are the model systems that can link in your case, through those Tribble proteins to the things that we know well, and you can identify the things that we can work on that by joining with collaborative teams around the world, we can hope to see advances that will lead to understanding for the betterment of treatments and performance of modern medicine. So I believe that you will all join with me and agree that Peter passed the test, that what we saw was something that met the criteria of outstanding performance of someone who is still at an early stage in his career, that has generated knowledge that in the rest of your career, both yourself and through the researchers that you will train and inspire, will go on and to make a real impact on the world. We look forward to seeing that impact be made, but I would like to ask you to join me in thanking Peter for just such a wonderful lecture before I invite the Vice-Chancellor to give you awards. So thank you very much, Peter. So now we have a table over here that has some things. I'll invite the Vice-Chancellor, Professor David Murdoch, to come and present the award. You're welcome to say some remarks if you like.
I think the physicist has summed it up very well.
So please, please come and receive the Carl Smith Medal. And there is wrapped a book that you may wish to, after the lecture, unwrap and show people what Carl Smith wrote. That is New Zealand from A to Z, from N to Z. And a third token of our appreciation. I did understand that someone had put up a poster of your talk on their bedroom door, but here's one that's framed that can go at a very special place in your home. With that, I thank you all for attending. There may be some official photographs being taken, so please thank you for coming and enjoy the rest of your evening. Ka kite anō.